video is Learning Target 8, Unit 7 on genetics, gene regulation. And in order to talk about gene regulation, we first have to talk about gene expression. We've actually been talking about this in Learning Targets 4 through 7. Gene expression is nothing more than when a gene is transcribed into RNA and then translated into a polypeptide or a protein. But only a fraction of genes are expressed at any time. Why is that? Well, take for instance the fact that you in your cells, every one of your cells has the instructions to make a new hand. Are you currently making a new hand? No. So you don't need those instructions. Those genes are not being expressed. Instead, they are being regulated. And so a cell regulates which genes are expressed in order to save energy and in order to control cell processes. This is going to be different in prokaryotes than it is in eukaryotes. In prokaryotes, they control the expression of genes by using mechanisms called operons. Here's a picture of an operon. I'm just going to draw this, of course. And so operons, this looks like the word operator. Well, it actually does have an operator in it. These are just groups of genes that regulate gene expression. That's the definition of an operon. Well, the most simple operons have two things in them. They have a section, the screen is DNA, it's just the gene at hand. They have a section called an operator, and the operator is the portion of the gene that is necessary for the gene to be expressed. It's kind of like the place where the enzymes go, like a promoter. All right. So here's our friend RNA polymerase up here. Let's draw RNA polymerase. And RNA polymerase normally can land here and whip across this DNA, make a messenger RNA. That messenger RNA will be made into a protein. Well, another piece of the operon puzzle is something called a repressor. And that's this red structure that I just drew. All right? And the repressor literally sits down on top of the operator and keeps RNA polymerase from expressing that gene. Well, I'm going to use the example of a particular operon in prokaryotes called the lac operon. And it's called the lac operon because it controls the expression of a protein that creates, or this protein that actually digests the sugar lactose. Bacteria aren't always in the presence of lactose, so aren't always needing to break it down, so therefore this protein is not always being expressed. However, when lactose comes in contact with the cell, so here's a molecule of lactose, that lactose will actually bind to this repressor, causing it to lift up. When it lifts up, RNA polymerase comes down, makes that protein digests all of the lactose until you have one piece of lactose left. That is this piece here. So it gets digested. The repressor sets back down on the operator, keeping it from being expressed yet again. And so when lactose is no longer present, the repressor binds again to the operator, preventing transcription, therefore preventing gene expression. Again, a very simple way of regulating genes. This happens in prokaryotes. In eukaryotes, it is much more complicated. This picture demonstrates some of that, and it's more complicated even than that. I could spend an entire class year talking about gene regulation in eukaryotes. We're not going to do that. We're just going to spend a few minutes. Much more complicated than prokaryotes due to many more genes and due to this concept called cell differentiation, which in itself is a kind of gene regulation. Cell differentiation is when cells take on different roles as the organism develops. All cells are different, so are A group of cells is different from another group of cells because it has a different function. There are cells in your body called stem cells, and these cells have a kind of a blank slate. They have the ability to become any kind of cell. They're typically found in the early stages of animal development, but they can also be found and as adults in the tissues in parts of your body that make new cells a lot, like the bone marrow and in your skin. Now, back to we'll go away from the crazy looking fly head for a second. There are kinds of genes in the, actually we'll go back to the fly head. There are some genes in the eukaryotic genome called Hox genes. And Hox genes control differentiation in multicellular organisms, and they do that by controlling the position and the location and the pattern of different parts of the body. They're active during development, and they are turned off typically once the organism is born. And so going back to this picture, you can see that a human being and the fruit fly have similar sections of Hox genes. This section always goes here in the head and the hands and the feet. You can see there's some different sections that humans have that fruit flies don't have, but fruit flies and humans have some similar ones. And if you manipulate those with a fruit fly, you can actually get legs to come out of their head, which is pretty fascinating or sickening, however you look at that, because these genes are very powerful. If you turn on, you can turn these genes on and off, and by turning genes off, that particular cell is going to express only a certain set of those genes. And so instead of being a nerve cell, for instance, it's going to be a muscle cell. Or instead of being part of your ear, it's going to be part of your spinal cord. And so if you take something like a stem cell, which has this blank slate, you can essentially, if you know what you're doing, tell that cell to become anything it wants to. And so that's part of the thrust in stem cell research is to gain an understanding on how to do this and how to regrow parts of our body that would be beneficial, like a spinal cord or some major organs. There are other genes that are regulated similar to Hox genes, and again, these are part of our embryonic development and are only turned on at certain times, like tissue repair. Like you break a bone, your bones aren't always being made, and so when you break a bone, your body has to turn those genes on in order to create that bone tissue. This can be done by cell signaling, and so this is just communication between cells. Cells receive chemical signals from the environment and from each other, and this allows them to differentiate or allows them to create new cells. You get a cut on your skin. The other cells around that say, hey, we need some new cells in here. The stem cells in your skin begin creating new cells, and those differentiate into new skin cells. And you could just imagine that going on in any part of your body as you're developing. And cells are touching one another. Certain cells are going to become part of your gut, and other cells are going to become part of your brain or whatever. And these cells are going to differentiate partly by their communication with one another and their position and location in respect to one another.